right. Good morning, everybody. Today is Monday, May 15th, 2017. And this is Before the First Cup of Jules here on UCY.TV. Happy Monday, everyone. It is a beautifully sunny 58 degrees, and it is going to get summery here this week in Western New York. So where do we leave off on Friday? We titled the show Probiotics, Microbiota, Living Therapeutics, DARPA, MIT, and Bill Gates. We spent a few days digging into gut bacteria and the microbiome and taking a look at some of the experimentation that has been going on. Lots of DARPA funded studies out of MIT, little company that spun out of some of that research that actually also got funding from the Bill and Melinda Gates Foundation, looking at ways to use bacteria to be able to reprogram your gut and produce pharmaceuticals. And we found a few different things in some of the research. I'm still working on looking a little bit deeper into some of the crazy stuff that we had ended up getting into by the end of the show. We were looking at this new type of bacteria that is showing up in a lot of commercial products called Bacillus, not to be confused with the Lactobacillus. This is a full separate branch of bacteria that includes anthrax, and there are quite a number of strains of Bacillus bacteria. And one of the things that's different about Bacillus from the standard bacteria that we know and love for gut health is that Bacillus family is a spore-based bacteria. That means it is dormant until it is activated in the right circumstances, in the right environment. And this Bacillus bacteria, certain strains of it, can produce antibiotics when it's inside of you. Those antibiotics can help wipe out quote-unquote pathogens or a bacteria that's already in your gut to then colonize with its new bacterial family. And in some of the things that I've been reading, there are quite a number of people much smarter than me that have spent a lot of years looking at microbiota that have been raising a red flag. And we stumbled across, of course, the spore bacteria by accident because I found it one time in a product that I purchased at the store. It was a tea. And I started questioning how, of course, probiotics could be alive when you dumped boiling water on them. And it kind of unraveled a big itchy sweater of things. And now I'm down this rabbit hole. Camille's been helping me research some of this, and she's been sending me some crazy links. And I got to tell you guys, this is much bigger than just being able to drop you some links. So I need to understand a little bit more before we dig any deeper into this. But I have to tell you, please, 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 if you purchase products with probiotics in them, especially if it's a product that you never saw it in before. You know, I always joke around and say Cheetos, any type of junk food. And there are a lot of different companies that are jumping on this bandwagon and licensing the use of these new founded Bacillus bacteria. And these are not newly found bacteria. So let me correct that. These bacteria they have found, I mentioned a case, CBS News published a story back in 2000 talking about this Bacillus bacteria that was discovered in the WIPP facility in New Mexico in the Carlsbad Caverns. Yes, WIPP, where they put all the Manhattan Project waste. Now, I don't know if they're actually putting waste there at the moment because they had a collapse. And they started moving some of that waste over to Andrews, Texas, in an above-ground facility for the time being, they say. But they found this Bacillus bacteria in salt in the Carlsbad mines that have been 250 million years old, and were able to bring it back to life. And that's the one interesting thing that got me to raise an eyebrow about this Bacillus, is that because it can lay dormant and because Bacillus is very resilient in being killed. They found out with that anthrax, like I said, that even irradiating it doesn't necessarily kill it. If you read up about Bacillus, that's the one thing that they say even chemical agents potentially will not kill this stuff. It's got to make you wonder if you let this stuff go in your body. And I did discover in 2 different articles the Bacillus bacteria is not a native inhabitant of the human gut. So this again is like an invasive species being introduced into a place where it typically wasn't. This is a bacteria that is found in soil. Sometimes farm animals will get infected with a bacillus, so that is where anthrax comes in. But something else more disturbing that both Camille and I ran across when we were reading documents over the weekend is the Bacillus bacteria, and I don't know if it's all of it. Again, like I said, I need to do a little more research and read some more on this, but at least some of the stuff that we were reading, Bacillus bacteria is a flagellate, meaning that it has a flagella and it can mobilize. And if you remember way back when, way back when, I'd say probably close to maybe 1.5 or 2 years ago already on the show, we had started looking at some of the nanotechnology and the quote-unquote nanobots and some of these little tools that they were already discussing and toying with in the lab that would allow them to target, let's say, cancer cells or some other site of disease with a biological or combination biological, technological device inside of the potential bloodstream or gut. They could then target disease or inflammation. And one of the little robots, quote unquote robots, like I said, whether it be some sort of little nanobot or a biological robot, because they do now have a programming language for cells, so they can tinker with them and make them do what they want. The flagellate was probably their favorite because it gave it motility. And they were able to use substances within the blood, and I don't remember what the one was. We read a study a while back of some things they were messing with, and it was using something within the human body as its battery cell. And then we'd read another story, one of the maybe it was George Church. It was one of the grandfathers of some of this technology had said, maybe it was actually Eric Drexler, in one of the interviews that we had watched, maybe even with Kurzweil, he had said that one of the rules that you don't break when you build these little things that go into the body is that you don't ever use something from the host to be a power source for an invasive organism that you're going to be placing into it. Of course, it makes sense. It is smart. But we discovered in some of the stuff that we were reading, Camille sent me a message going, oh, did you see that one of these Bacillus bacteria actually uses calcium to create the spore that then allows it to replicate itself. So it would be using, and I don't remember which Bacillus, there were quite a number of them, but one of the cautions we had also found, these different families of Bacillus, some are innocuous and some are extremely dangerous. There was one that could actually cause sepsis. And one of the biggest concerns was that once this bacteria had infested your lower gut, that once it created spores, it goes between a couple different states. One is a vegetative state when it is growing and alive, and one is the spore state. Once it reproduces, the spore is kind of like the egg. But once it's in that vegetative state, it has this flagella, and it can start traveling to different parts of the body. And they had even listed like the spleen, your lymph nodes, and ultimately in your blood. So I got to tell you guys, look at the label. If something is listed as a bacillus that you think is a probiotic that is healthy for you, I'm not going to say, like I said, it's not healthy for you, but there are other people out there that are raising the same red flags, the lack of safety research. Now, I said on Friday that I could find no studies. Well, this weekend I did some deep searching, and I actually did find some studies. I found a couple that were on independent, quote unquote, independent research publication websites. And it looked legit, saying, of course, this is great, blah, blah, blah. But one of the things you got to worry about is how long has this research in the human body been going on? How detailed was the research? I mean, the human genome was only completed in the late 90s, let's say 97, 2007, 2017. So we're only talking 20 years. But I don't think so. We are talking maybe 20 years that this research was potentially possible at the level that we have now. But where are the long-term studies on this? Where are the independent Food and Drug Administration research? I saw that they were marking this as believed to generally be safe, the Food and Drug Administration, when they rubber stamp this. And one other thing I mentioned on Friday is that some of these new Bacillus probiotics that they're spinning out there are going crazy all about it's non-GMO. But I remembered something after the show on Friday. I'd said, well, you know, you could hybridize things, put two bacteria next to each other in a petri dish, let them cross-pollinate, whatever they do. Kind of the way, you know, old school farmers would create their own breed of corn or whatever before the days of splicing and dicing, genetic modification. But one of the things that I remembered is that CRISPR technology, that would be some of the technology that Georgie Porgy Church and his group won the patent award against Jennifer Doudna at Berkeley for. CRISPR technology allows them to flip switches, turn things on and off, splice and dice. And it was ruled that if you CRISPR something, that does not necessarily constitute genetic modification. Now, I would argue with that, regardless if you're going to be putting foreign DNA into an organism or if you're just flipping a few switches. I mean, any intelligent thinking human would probably agree with me that, you know, that's 6 of one and half dozen of the other, you know, just different ways to say it's genetic modification. But according to the quote unquote law, CRISPRing does not necessarily constitute a genetic modification label. So they can tell you all day long that this new Bacillus bacteria that they are going to be putting in and calling probiotics is non-GMO and non-GMO verified. But remember what the legal definition of GMO is, and that is putting foreign DNA into an organism. Hey, you can flip a few switches and change a few things and make it create some sort of substance. Remember, they have been putting a lot of money from DARPA funding into messing around with cells at the Weiss Institute. Again, something Georgie Porgy Church is involved with. Turning them into little biofactories where they can have things such as E. coli create chemicals and biological substances that they maybe originally had created in a laboratory. So, you know, then we look at that living therapeutics, and that's exactly what they're talking about doing, is reprogramming bacterial cells to be able to produce quote unquote therapeutic chemicals when it detects that you have some sort of problem. Now, one other thing that I wanted to talk about when I was doing my research after the show on Friday, to put the notes underneath the show in the description. And by the way, anyone who may be new to the show or maybe never realized, if you hit before the first cup on YouTube or any third-party site that rips the show, from what I've seen, most of the places that are ripping this show are actually also taking the description of the show. So make sure you look in the description. I try very hard, but I talk about a lot of things, so sometimes I miss stuff. But I try very hard to go back through the show and link to sources for all this mad science that I talk about. So when you're trying to understand what I'm talking about, or when you're trying to share some of the craziness that I talk about with friends and family, you'll at least have a way to give them something to chew on so they don't look at you like you should be wearing one of those tinfoil hats. Go out and buy you one on Amazon. Last time I saw, they were all sold out, by the way. It's pretty funny, $14.99 for a handmade tinfoil hat. Anyways, when I was researching to do my notes on Friday, I ran across a story that I totally missed when it came out in 2014, but I wanted to share with you. This also came out of the Weiss Institute, again, a collaboration between Harvard and MIT that has George Church and DARPA working all together, titled Bacterial Reporters That Get the Scoop. Engineered bacterial sense, remember, and report on their experience in the gut, paving the way to living diagnostics and therapeutics. So this even goes one step further than just living therapeutics. They are talking about doing living diagnostics and therapeutics. This is out of Weiss Institute's website, wyss.harvard.edu, March 17th, 2014. It is a jungle in there, in the tightly woven ecosystem of the human gut. Trillions of bacteria compete with each other on a daily basis while they sense and react to signals from the human immune system, ingested food, and other bacteria. Problems arise when bad gut bugs overtake friendly ones, or you take some spore-forming bacteria that just overtakes your gut. I mean, honestly, guys, I wanted to point this out, if you look at the ingredients on any of your substances that you're ingesting that supposedly have probiotics, and you see something that is titled just Bacillus something, or B dot something, look it up and make sure it is not one of those spore-forming bacteria. Until we dig a little bit more into this, I'm going to warn you, flags of caution. Okay, there is definitely something odd about this whole thing. But anyway, back to this. So when problems arise, bad gut bacteria a lot of times overtakes friendly ones, or when the immune system is thrown out of balance, such as in Crohn's disease. And remember, your gut bacteria has a lot to do with your immune system and can influence many different things, including brain chemicals and that vagus nerve that direct connects from the gut to the brain. Doctors have struggled to diagnose these conditions early and accurately, but now a new engineered ding-a-ling-a-ling strain of E. coli bacteria could deliver status updates from this complex landscape to keep the gastrointestinal diseases at bay. This new strain of non-destructively detected and recorded an environmental signal in the mouse gut and remembered what it saw. This advance, reported in the proceedings of the National Academy of Sciences, could lead to radically new screening tools for human gut health. Now, if you remember, we just found out that Eric Dishman, the director of the National All of Us program, and this is this precision medicine initiative to provide precision medicine to all. This is to get people to give your DNA swab, step right up, get into the database so they can splice and dice our data to be able to learn more about how humans are constructed. And this is at all different levels. This bacterial reporter and these living therapeutics and our doctor's smartphone back-ended to artificial intelligence and diagnosing mental disease, all by the sound of your voice. Tommy Insel, former director of the National Institute of Mental Health, now at Google's Verily division, and all these tech companies and DARPA getting into bed together to help bring us this 21st century of wonders medicine that'll be able to bag and tag every bit of us, is all coming together quite nicely and making a boon on Wall Street for investors. The key to turning E. coli into gut reporters, as they call them, to be able to surveil you, was to insert a well-known genetic switch that flips it, a bag tag switch flip, when it senses a specific environmental cue. This switch confers on the cells the ability to remember what they sense for up to a week, long enough for scientists to recover fecal samples from you. And Google wants to collect your poop, remember, and test whether the switch has been flipped. This achievement paves the way toward living monitors inside of you, programmed using synthetic gene circuits, said Weiss Institute core faculty member Pamela Silver, PhD, senior authority on the study, who is also the Elliott T. and Oni H. Adams Professor of Biochemistry and Systems Biology at Harvard Medical School. Silver's team includes James Collins, PhD, who's also a Weiss core faculty member and a professor of bioengineering at Boston University, as well as other collaborators from the Weiss Institute, Harvard Medical School, and Boston University. This could lead to brand new diagnostic tools for all sorts of complex environments. Dang, maybe not just humans, maybe the soil as well. And I remember Dr. Huber talking about some new life form that he had discovered in the soil that had been treated with glyphosate that killed all of the beneficial soil bacteria. Some of them could have even been some sort of Bacillus family. And they had been invaded. The soil itself had been invaded by some new organism that wasn't bacterial, wasn't viral, nor fungal. And he could only term it as a brand new life form. But hey, if they were flipping switches of bacteria in the lab and then maybe testing it out, or hey, it escaped on somebody's shoes and got into the soil and found an opportunistic capability to grow and maybe even kill some of the native bacteria, that could account for some sort of weird new thing that he had never seen before. I don't know, just tossing that idea out there. But, this has got to be frightening, guys. When you start thinking about Clapper going out in front of the Senate Intelligence Committee and claiming that CRISPR should now be considered a WMD, and we are able to now CRISPR the planet, anything that is biological, they can take the DNA and start flipping switches left and right just to see what it would do. And the controls on this stuff really have not been put into place. We have, I had a story, and I don't know, I'd have to find it because it's not up here right now. But there was a story on Friday I ran across that it talked about how even our laws are so far behind scientific breakthrough and how fast it is happening at this point. We really are in quite a conundrum. And as this technology from all different points and all different realms is being utilized, it is increasing exponentially, not just in its reach to, little Johnny and mommy's back bedroom, but also in its scope vertically. It is being able to target every form of life on this planet, not just human, not just animal, not just plant, not just insect, not just soil. We are getting to realms where they can bag, tag, cross, integrate. Remember the DARPA lady had said that they are hoping at some point in the future to be able to just push a button and create life from across every type of known sequence. Create life on the fly, to be able to be used as a utility to help them create things or fix things or produce things. Living biofactories is where it is at, and this Weiss Institute is one of the organizations that are helping them just do that very thing. The genetic switch the team inserted into E. coli came from the lambda phage, and phages are something that is one of those organisms or technologies that I don't know enough about, but it is very important. It's come up in a lot of different things. I really, really need to spend some time researching the phage and the cells and viruses and how these can interact and cross-pollinate. One thing I want to mention to everyone, I know I've mentioned this long before, but I am going to put a call out again. There is an open courseware project being put up by MIT, ironically enough, the same group that is involved in a lot of this mad science, but they are offering what they are calling open courseware, and that is a lot of their classes free and available online. A lot of the classes have video as well for lecture. They are offering a lot of the material and the class material online, as well as notes and labs, and they are also linking, if there is a textbook that is involved, to Amazon, where you can then get the name of the textbook and then search for it if you want. I was able to find the biology syllabus for the biotechnology concentration at MIT. I know I was saying on Friday I should go back to school for biotech, but hey, why even bother to have to go sit in a classroom when you can get it online? So biology first year, and I did take it in college, but I mean, it's been 20 some odd years. I'm sure things have changed. We've learned a lot more. So probably be recommended again, and a refresher for stuff I've forgotten. But there is a biology course, 101 or whatever they call it, 107, that really starts looking into all the systems of the cells and how they interact and the different proteins and power sources. And I am considering working on this little by little. If anyone is interested, the class said it took about 150 hours, so God knows where I'm going to find 150 hours, but I sure am going to try giving it a shot, even if it's just an hour a day, maybe late at night. I found the textbook on Amazon for it was just shy of $9 used, so I think I might even buy that and use it for reference. But there are videos and everything out there, so if anyone's interested, let me know. I'll give you the link. I will put it in the notes. It is ocw.mit.edu/courses, plural, /biology. Check it out. There's a lot of stuff. I'm sure if you back that off to just the ocw.mit.edu/courses, you will probably find a lot of their open courseware. But anyway, back to this. So they are using phages. I ran across phages when they were looking at genetically modifying bacteria to then go in and infect things to do genetic modification. It was back in some of that Monsanto technology. So the fact that they are using phages in E. coli to then make some changes and then put them into a living organism to do different things and flip switches is very disturbing to me. That is screaming genetic modification of humans through our gut without our knowledge. So this says the genetic switch the team inserted into E. coli came from a lambda phage, which is a virus that commonly attacks this bacterium. But after invading E. coli, lambda typically lays low, living in a stealth mode called lysogeny, in which its DNA simply just hangs out in the E. coli's genome. So it's just kind of like a innocuous infection. But hey, don't worry about it, because they can go in and do things to it. But when this bacterium's DNA is damaged, the switch flips, instructing the virus to enter a mode called lysis, in which it multiplies inside of the cell and then breaks through its membrane in a kind of microbial explosion. Again, another invasion of the body snatchers. This is a very stable system in nature, says lead author Jonathan Cotola, PhD. We knew the lambda switch would be a very great candidate for the memory element, and we simply tweaked it to meet our needs. cells engineered with the lambda switch would not be lytic under any conditions. Cutula and the rest of his team used standard molecular genetic tools to rig the switch. They're going to rig switches. Like, this is just so horrifying to me that it turned on only. And where did we hear this before? It turned on only in the presence of an inactive form of the antibiotic, ding ding, tetracycline. Oh no. Remember, the tetracycline switch was also used by Oxitec in those genetically modified Zika mosquitoes. And then there was question as to whether or not maybe they had a little problem on their hands releasing those mosquitoes, because tetracycline seems to me a lot more prevalent in the environment than they thought. It is used in livestock and in other things. It's given to people. It ends up in the water supply. Tetracycline may not, I don't know about this researcher, but it may not be such a good thing to make it turn on genetically in the presence of tetracycline. Of course, in laboratory experiments, they can isolate it. The switch turned on within a few hours of exposure to this antibiotic, and it stayed in this on state inside of the E. coli for a week or more, even as the bacteria grew and divided. In short, the cell remembered what it had seen as a molecule in the gut. So tetracycline turns on a little microbial E. coli recording device inside of the gut of a living organism, and it is turned on via tetracycline. Yeah, that is a summation of what we just read. What? It's crazy. So wait a minute. So some of the spore, so let's think about this for a second. Some of the spore Bacillus that we've been reading about, and Camille sent me a couple of links to videos and some of this new branded Bacillus bacteria, not E. coli. Bacillus is the spore forming. Some of this Bacillus spore forming bacteria that Camille sent me this video about, they put a brand name on it. They're selling it as a brand new probiotic. Oh, it's going to keep you healthy, make you live forever. No, not really, but they're really pushing this health thing. It can also produce 25 different, and they do not tell you which those 25 different antibiotics are inside of the gut. Wouldn't it be interesting if one of those 25 happened to be tetracycline? I started thinking also E. coli. They've been doing a lot of experimentation on tinkering with E. coli, and this is just one of many different experiments with CRISPR and different infecting agents. E. coli, though, if you remember a while back, we saw E. coli come up quite a bit in the news. You had packaged lettuce and strawberries. Then you had the whole chain. I don't remember which chain it was. Was it Chili's maybe? Chili's had said that they were going to start using non-GMO foods in their restaurant products. And then they had quite a number of outbreaks of E. coli, where large numbers of people had gotten sick. And there were claims at the time that it might have been a form of quote-unquote bioterrorism done at a corporate level, that someone was sabotaging them. That was what they kept claiming. And, there have been stories way back, even after 9/11, where they were really kind of pushing this bioterror mantra. There were some anthrax attacks. Again, anthrax, a bacillus, where, there could be attacks on the food supply. They had talked about salad bars as being a potential, being able to aerosolize some of these different bacteria. I'm just saying, now that we know that E. coli has been studied so extensively in the lab that they can flip switches and make it do stuff and insert phages and be able to overtake its body and create a virus, essentially, that can then produce things that can rival gut microbes inside of a living organism. It's just got to make you wonder if E. coli infection is natural, or if it maybe was some sort of human experimentation gone live. I don't know. Calling me tinfoil Hattie, but I'm just saying, seems a little suspicious to me that E. coli is one of those organisms that are so prevalent, and we have had a few odd little outbreaks there. It does occur naturally, so I'm not going to say, but just saying. But anyway, but to function as a living diagnostic, this E. coli also had to survive their trip through the gut intact, which meant that they had to compete effectively against rival gut microbes. Ding, ding. This engineered strain worked fine in laboratory experiments, but gradually disappeared when the team introduced it into the gut of the mouse itself. It turned out that it had been outcompeted by the animal's native gut bacteria. The team did not fret in the face of this result, though, because they knew that the classical strain of E. coli that they had used only lived in the laboratory since 1940, losing its ability to compete in the real world, particularly in an environment as challenging as the mammalian gut. Well, phew. So they used one that couldn't compete. But wait, don't worry about it, because they got over that. They tackled this very problem, so it now could live inside the gut by isolating a native strain of E. coli from the mouse gut. And then they engineered this E. coli and its genome to incorporate that same switch. The switch in the mouse E. coli flipped within hours as it had before, and the cell, quote unquote, remembered for about a week that they had seen the antibiotic in the gut. Moreover, the population stabilized within the gut, holding its own in the presence of the native gut bacteria. This team now envisions a day, and remember, this was 2014, and they've gone a lot farther than this in the lab. This team envisions a day when a doctor, ding ding, would give a patient, ding-a-ling-a-ling, a strain of engineered bacteria as a diagnostic tool, much as they give probiotics today. Hello. The strain would be rigged and engineered to monitor the gut for any number of conditions, from inflammation to disease markers. They probably could even tell what you ate for lunch and what illicit substances you have ingested. Those were my words, but it's true. The strain would be rigged to monitor the gut for inflammation and disease markers, and at a follow-up visit, the patient would submit a stool sample, or they might have a smart toilet. Remember, we looked at those. Smart toilets are coming as well, and Google wants your stool sample. And then medical technicians would collect the E. coli from your sample and analyze it. And DARPA is also working on the fly diagnostics for the battlefield, of course, but they could also put that into your smart toilet arsenal of tools. Only if the switch or switches were on would doctors perform the more invasive tests, oh no, such as an endoscopy or a colonoscopy. For now, the team is focusing on genetically tweaking the memory element of their systems so that the cells remember for even longer periods of time, longer than a week, and engineering it so the switches flip when it senses other chemical signatures as well, such as when it finds cancer or parasites or, in the future, illicit substances consumed. In the longer term, their engineered bacteria could sense a disease state and then work with other engineered genetic circuits that you also ingested, maybe in your probiotics, that could produce a synthetic drug on demand, thus producing a dynamic therapy within the organism. Again, little living creatures inside of you, providing diagnostics and drugs to cure, quote unquote, cure you. They could even tamp down on Johnny getting a little bit excited there. It's not too much of a stretch to have it also start looking at other biological features that maybe indicate that you're having a stressful moment. Let's get that bacteria producing the chemical signatures that stimulate that vagus nerve, that produce oxytocin to calm Johnny down and make him trust us more. Just saying, guys, but it definitely is not out of the realm of science now. We have talked about all these different things. They are working on all of it, and they have found ways to manipulate all these different biological processes. Again, chinks in the armor. As they discover more and more, we are more at risk. And these little micro machines are becoming smarter and smarter, and they can program them and then just put them simply into our food. Now with probiotics, wink. Our increasing appreciation of the role of the microbiome in health and disease is transforming the entire medical field. The concept of using the power of synthetic biology to harness microbes that live inside of our gut to develop living diagnostics and therapeutic devices is a harbinger of things to come. And yeah, that's a great word choice. I would have probably chosen that very word myself, a harbinger. And the scientists' work provide the first proof of principle that this is a viable and exciting, says the author, actually says the founder of the Weiss Institutes itself, Don Ingeber. It's a very exciting path to pursue. And the last little footnote at the bottom of this story says this work was funded by DARPA and the Weiss Institute. So put that in your pipe and smoke it. Because if there ever was writing on the wall or any type of verification to all this craziness that we've been talking about on this show, there it was, directly out of the harbinger's mouth. So on top of being able to body snatch our gut, to be able to monitor, surveil, and quote unquote cure, and manipulate you, we also have talked about smart dust and neural dust being developed by DARPA and Berkeley that can be implanted into your nerves and into your brain. We read about to monitor you, sand size, sensor implants to give instant feedback from nerve cells. They are calling these electroceuticals. We found out that Google also partnered with GlaxoSmithKline to work on electroceutical devices and different types of neural dust. It is coming, people. The approach that they are taking is ingenious, says neuroengineer Jacob Robinson. They have addressed one of the most important challenges when it comes to neural interfaces. So scientists have previously explored using radio waves to power and communicate with neural dust motes. However, radio waves are not good at reaching deep within the body, While decades of ultrasonic imaging have revealed that ultrasonic pulses are very good at penetrating tissue, they were using ultrasound to generate electricity with piezoelectric sensors on these little dust motes. Experiments so far with neural dust motes have only involved the peripheral nervous system, which serves the limbs and your organs, but not your full central nervous system, consisting of your brain and your spinal cord. Still, electroceutical therapies may still have many applications in the peripheral nervous system, such as bladder control or appetite suppression, as well as many other things. In the long run, scientists want neural dust motes in the brain and spinal cord, but their one challenge is targeting the central nervous system because ultrasound does not pass through bone. They are also looking at creating brain-machine interfaces, so that's not impossible by any means. But researchers are now working on miniaturizing even further these dust motes and discovering more biocompatible materials to package them, so of course they last lots longer in the body, as well as adding more sensors into a single dust mote so they could be used anywhere in the body, not just the nervous system. Guys, I've talked about many times us becoming a part of the Internet of Things, soon to become the Internet of Everything, with us as just a simple node on the network. We've talked about that neurological surveillance system that is cited in 399S in the 21st Century Cures Act passed by Congress in December of 2016. This is not a fantasy, people. They really are talking about this. They are talking about big data to cure disease. And of course, like I said, be afraid when they are crying about the costs of healthcare, because this is going to step up, even if they're not talking about it now. They are lining up all of the dominoes to be able to tell you that this is going to be the way that they're going to fix us. They're going to bring the cost down. They are going to make it smarter and better. They're going to make us live longer. With 21st century medicine, we need to have a dashboard so they can monitor every function inside of you and be able to intervene before disease has a chance to progress. Now, I am not saying, like I've said many times, I am not saying that the technology itself is evil, but the ones who are owning the technology have a great potential to use this for, yes, good and for evil. Once this is implanted inside of all of us, and remember, a lot of these things are so damn tiny, we might not ever even know that they are inside of us. You know, some sort of spore-forming bacteria that maybe has had some switches flipped inside of it that we would never know. Maybe some E. coli that we ingested in a yogurt, let's say. Maybe some sort of dust mote that was so damn tiny and we drank it in some bottled water or ingested it in some sort of nutraceutical vitamin. I don't know. Who knows? I mean, a lot of these things, a lot of these supplements especially, say that they're not even FDA approved. It's got to make you wonder who is behind all of these different products. And I'm not going to say again that there is insidious things going on, but all it would take is one. All it would take is one line out there or one manufacturer that then gets a label saying that it's safe or it's not genetically modified or that it's organic, wink, let's say, and then run it into lots of product lines out there that lots of Americans especially consume, to then be running a big covert operation like Google wanting to collect your poop. Or smart medicine in the future demanding you have a smart toilet. Maybe we don't know it, but maybe you have smart microbes already in your gut that are reporting on you. They just aren't decoding that information yet. I'm just saying, guys, it could be so covert that we could have already been compromised long ago. This research is being published now, but remember, they always publish things years after the research initially started. Story came out of Open Mind, which is a publication that has some connection to MIT Technology Review because they actually shared this. Titled, The Fight Against Cancer with Artificial Intelligence and Big Data. And remember, cancer is really kind of the first dangling carrot where they are getting people to step right up and get used to little nanobots in your blood to target quote-unquote disease. They are promoting that all over. You're seeing that on mainstream TV now. Just about anyone you talk to doesn't think you're crazy when you talk about using nanobots to target cancer because they promoted that enough. But everything that we were talking about with IBM Watson being now an oncologist and, being able to build the database that he can access 24/7 and compare different cases is becoming status quo now. And that fight against cancer is going to become the fight against all disease eventually, with the back end of AI that becomes Dr. Smartphone and the big, huge pulsating database, starting out with something like the All of Us program here in the United States to, yes, not only diagnose you, but to also give them all the chinks in the human armor. From anywhere, with just a mobile phone, the story says, May 5th, 2017, anyone can become an air traffic controller or at least a virtual air traffic controller. One can follow the world traffic flow of airplanes live and find out where an aircraft is coming from and where it is headed. One just has to take advantage of the millions of pieces of data that fly daily across the internet. This is the magic power of big data, and artificial intelligence then enters the picture to find patterns and give meaning to the massive and heterogeneous information stream. Together with these two technologies, they have embarked on a colossal mission far removed from their usual commercial applications, and this would be to now find treatments for diseases such as cancer. When MIT professor, ding-a-ling, again, MIT, Regina Barzilay was diagnosed with breast cancer, she not only lost her personal life, but it also turned her research upside down. In the endless shuffling between hospitals, she realized that most of the information being recorded about patients was not being used. Clinical decisions are often based on clinical trials, and that is 3% of the patients participating in them. That means that the entire experience of what happens to 97% of patients is not being used. How would Amazon work if it discarded 97% of its data? I think we are sitting on a gold mine of data that we are not using, she maintained. Ding-a-ling-a-ling-a-ling-a-ling-a-ling. See, up until this point, we had silos, that's what we called them in the corporate world, of data sitting all over. And that was data that if you had a human look at it, maybe it would become actionable. But if it was scattered all around the globe and on different people's computers and in different publications and applications, it would be very difficult for a human to collate all that data. We found out that's what Watson was doing. Watson went out and scanned the internet to find any references to the same sorts of cancers, let's say, or clinical trials, to be able to then collate all that data and put it into one big place that then it could analyze for any commonalities between them and potentially find a cure. Guys, this isn't science fiction anymore, and this is something that they are going to be rolling out to all of us per Eric Dishman, the director of the U.S. All of Us Precision Medicine Initiative. Oh, and the story I was just reading closed. I love when that happens. Anyways, they're teaching machine language, blah, 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 and it can go on and on. But genetic big data is one of the most important discoveries that the extensive decade-long research into cancer has brought us. And it's not just one disease, but many different. There is no single liver cancer or no single type of pancreatic tumor. The origin of the cancer in each patient has its own causes. And this is where the medicine of precision is about sequencing and the analysis of the genome. Step right up and provide your DNA swab for the future of precision medicine. Guys, this is where it's going. And so now we're even moving a little further. Drug discovered by artificial intelligence. There is a new company out there that has developed a new anti-cancer drug against pancreatic, breast, liver, or brain called BPM 31510, which was discovered using an AI algorithm. They have collected samples of cancers and healthy tissue from 1,000 patients. It processed all the data with AI, which then proposed a treatment. We have now essentially reversed the scientific method. We allowed biological data from patients to lead us to a hypothesis, and now they are beginning clinical trials to test the drug's effectiveness. So there you go. Drugs by AI. In addition, major technology companies have already begun applying big data and AI to health. Alphabet is using it to collect samples of thousands of healthy volunteers, and that's your poop, as well as Apple is taking advantage of data from millions of iPhone users with the Research Kit and Care Kit, and Microsoft is developing tiny sensors that can be worn on the skin to transmit your biometric data to remote health monitors. All right, guys, have an awesome